Essentially, all hormones are variations of one of two chemical types, peptides and steroids. The mechanisms of action are quite different for these two classes of hormones. The peptide group binds to their target receptors on the plasma membrane. Receptor-ligand interaction initiates an intracellular signal transduction cascade that may involve second messengers such as cyclic AMP, cyclic GMP, and IP3, or tyrosine kinase activity either intrinsic to or associated with the target receptor. On the other hand, the typical site of action for the steroid group, as well as thyroid hormone, is the cell nucleus. The key thing to remember is that steroid hormones, such as testosterone, are lipophilic. There are two important consequences of this chemical feature. First, steroid hormones can pass freely through cell membranes and into the nucleus. And second, they are insoluble in plasma and are carried by specific molecules called binding globulins. Steroid hormones pass through the lipid bilayers that compose cell membranes and interact with receptors, either in the cytoplasm or nucleus, which then adopt a conformation able to bind DNA to alter gene expression. Thyroid hormone has a similar mechanism of action as steroid hormones, and indeed, there is a shared family of steroid and thyroid receptor molecules. As you can imagine, since these hormones act by altering gene expression, their effects take time to manifest. Contrast this to the rapid effects of a peptide hormone such as insulin, which causes an immediate increase in glucose uptake by cells, enabling cellular respiration and ATP production. Binding globulins increase the solubility of the steroid hormones they carry and improves delivery to target tissues. The serum level of binding globulins often has clinical relevance. For example, sex hormone binding globulin, or SHBG, is the specific carrier for sex hormones such as testosterone. Increased levels of SHBG lower the amount of free testosterone. In men, decreased free testosterone manifests as gynecomastia. When the levels of SHBG drop in women, there is more free testosterone in the bloodstream, which manifests as hirsutism, which is male pattern hair growth. Let's move on to discussing thyroid hormone. The pathological conditions of hypo and hyperthyroidism are absolutely critical to know for the USMLE. Thyroid hormone is produced in follicular cells, also known as thyrocytes, of the thyroid gland. It contains iodine and controls the body's basal metabolic rate. You can see in this diagram of a follicular cell that thyroglobulin protein, TGB, is produced in the cytoplasm and secreted into the lumen. In parallel, iodide ions are taken up by follicular cells via a sodium-iodine cotransporter, oxidized to diatomic iodine, and then secreted into the lumen. In the lumen, thyroid peroxidase is responsible for adding iodine onto tyrosine residues of thyroglobulin. Upon stimulation by TSH, iodinated thyroglobulin is endocytosed from the lumen back into follicular cells, where it is digested by lysosomal proteases to release monoiodotyrosine, MIT, and diiodotyrosine, DIT, into the cytoplasm. Combining MIT with DIT results in triiodothyronine, known as T3, whereas combining two DIT molecules results in thyroxine, known as T4. These two forms of thyroid hormone are then transported across the basolateral membrane of thyrocytes and into the bloodstream in a TSH-mediated process. Thyroid peroxidase is a versatile enzyme. Not only does it incorporate iodine into thyroglobulin, a process known as organification, it is also responsible for oxidation of iodide ions, and coupling MIT and DIT to form thyroid hormone. It's worth pointing out that T4 is the major product of follicular cells. Though some T3 is produced in the thyroid gland itself, the majority of T3 in the body is derived from peripheral deiodination of T4, specifically a removal of the 5' iodine by 5' deiodinase. You can consider T4 to be a prohormone for T3, since T3 is much more active than T4. Okay, let's turn now to the functions of thyroid hormone. As we already noted, the major role of thyroid hormone is to control the body's basal metabolic rate. How does it do that? In brief, thyroid hormone increases sodium-potassium ATPase activity and expression, which increases oxygen consumption, the respiratory rate, and body temperature. It also upregulates glycogenolysis, gluconeogenesis, and lipolysis to increase ATP production. Additionally, thyroid hormone upregulates the number of beta-1 receptors in the heart, which leads to an increase in heart rate, stroke volume, contractility, and therefore cardiac output as well. This is the reason why hyperthyroidism is associated with palpitations. There are two other critical functions of thyroid hormone in the developing body. It promotes bone growth along with growth hormone in a synergistic fashion, and it promotes maturation of the central nervous system. Patients with thyroid deficiency early in life develop neonatal hypothyroidism or cretinism, which is characterized by severely stunted physical and mental growth. For example, short stature and low IQ. You may find it helpful to think of thyroid hormone in terms of the 4 B's: brain maturation, bone growth, beta-adrenergic effects, and increased basal metabolic rate. Like steroid hormones, thyroid hormone is transported in the blood by a binding globulin, thyroxine binding globulin. TBG binds most of the T3 and T4, and the only thyroid hormone that actually acts on the tissue is the fraction of free, unbound thyroid hormone. Lab tests are available to measure the total amount of hormone as well as the free portion of the hormone to allow clinicians to understand the source of changed hormone levels. Two situations in which this is relevant are in hepatic failure, when the TBG level is low, and pregnancy, when the TBG level goes up. So in pregnancy, for example, the TBG is high and the total amount of thyroid hormone is high, but the amount of free hormone should stay constant, and therefore the patient is considered to be euthyroid. Make sure you don't confuse TBG, or thyroxine binding globulin, a carrier of thyroid hormone in the blood, and TGB, or thyroglobulin, the precursor of thyroid hormone. Graves' disease is an autoimmune condition in which production of thyroid-stimulating immunoglobulin, or TSI, in large quantities stimulates follicular cells in a similar manner to TSH, resulting in hyperthyroidism. Two common antithyroid drugs used to treat hyperthyroidism are propylthiouracil, or PTU, and methimazole. While PTU inhibits both thyroid peroxidase and 5' deiodinase, methimazole only inhibits peroxidase. Both drugs take time to have a clinical effect because they block synthesis of thyroid hormone but do not deplete existing stores. Interestingly, excess ingestion of iodide can lead to a transient decrease in thyroid hormone levels because of thyroid peroxidase inhibition. This phenomena is known as the Wolff-Chaikoff effect. Okay, before we leave endocrine physiology, let's do a practice question. A 17-year-old girl comes to see you with the chief complaint of primary amenorrhea. After physical examination and additional testing, you discover that she has a blind-ending vagina, no uterus, cervix, or ovaries, as well as undescended testes in the inguinal canal. She also has a 46XY genotype. What is the most likely diagnosis? The patient has a Y chromosome, and therefore produced sex-determining region Y, or SRY, and Müllerian inhibiting factor, or MIF, in development, which you'll learn more about in the reproductive lectures. MIF blocks the development of female internal reproductive organs, and SRY stimulates differentiation of the primitive gonads into testes. This phenotype-genotype combination points to androgen insensitivity disorder, a condition in which androgen receptors are absent. What would you expect the patient's testosterone levels to be? Testosterone would be very high because without androgen receptors to sense the circulating testosterone, there is no feedback inhibition on the production of LH, and the body stimulates testosterone production as if there is very little around. If there is one thing you should take away from this lecture, it is that endocrine physiology is all about feedback loops. Usually, negative feedback is involved to maintain homeostasis. However, don't forget that there are some important exceptions in which positive feedback is involved, such as the LH estrogen loop that triggers ovulation.